Let's see with the locomotion in Proctoctista and invertebrates. There's an immense variety of organisms with different models of locomotion. Locomotion in Euglena. Euglena moves with the help of flagellum. As the flagellum is whipped backwards, the organism moves forwards. However, when the flagellum moves forward, Euglena does not move backward. Locomotory flagellum is an anterior end of the body and pulls the organism forward. Waves of activity are generated by the flagellum itself and they pass in a spiral fashion from its base to its tip. They increase amplitude and velocity. The activity node of the flagellum causes the body of Euglena to rotate forward about its axis. Euglena is able to change its direction by the active contractile myonemes which run along the length of its body. When they contract, the shape of the body as well as its direction changes. First the body becomes short and wider at the anterior end, then in the middle and later at the posterior end, and this characteristic movement is called the euglenoid movement. Locomotion in paramecium. Paramecium moves with the help of cilia and is called the ciliary movement. All the cilia do not move simultaneously, but a bunch of cilia move in a progressive wave-like manner at a time. The wave starts at the anterior end and progresses backward. Cilia are short, fine thread-like extensions of the cell membrane, and the length of cilia ranges from many microns to 100 microns, and the diameter varies from zero point 0.1 to zero point 0.5 micrometer. A cilium consists of 9 peripheral double fibrils, giving the appearance of egg-shaped figure and 2 central smaller fibrils. All these fibrils run longitudinally through the cilium. These are covered with the extensions of the membrane. The exact mechanism of movement of cilia is not known. However, in 1955, Bradford suggested that movement of cilia is due to the simultaneous contraction or sliding of double fibrils in two groups, one after the other. Number 1. 5 out of 9 double fibrils contract or slide simultaneously with the result that cilium bend or shorten, called the effective stroke. The 4 out of 9 double fibrils contract and cilium becomes straight. It is called the recovery stroke. As a result of bending and recovery stroke, the paramecium swim against the water. The energy for the movement of cilia is provided from the ATP. The enzyme present in the cilia breaks up the ATP to release energy. The action of the cilia is coordinated and all the cilia beat together in a sequence to propel the animal in one direction. Locomotion in amoeba. In amoeba, movement takes place by the means of pseudopodia. Pseudopodia are the finger-like projections thrown in the direction of flow of the cytoplasm. Consequently, the body moves in that direction. The exact mechanism of formation of pseudopodia is still debatable. Locomotion in the jellyfish. Jellyfish has an umbrella-like body called bell. First of all, water is entered in the bell, then the bell contracts, the water is forced out like a jet and the animal moves forward. This movement is called the jet propulsion.